Hello friends, welcome to another video. In this video, I will explain the chemical constituents of DNA. DNA is made up of a monomer called nucleotide. What is nucleotide? Nucleotides are the building block or monomeric unit for both DNA and RNA. And the nucleotides are made up of nucleoside and a phosphoric acid. Nucleosides, what is nucleoside? Nucleosides are the component of nitrogen base and sugar. There are two types of nitrogen base is found in both the nucleic acid DNA and RNA. In chemical nature they are different. So there is two type of nitrogen bases, nitrogen containing bases. There is purines and pyrimidines. Adenine and guanine are the purines and the thymine, cytosine and uracil are the pyrimidines. And the two types of sugar is found, both DNA and RNA. In case of RNA, it is ribose sugar, or in DNA, it is deoxyribose sugar. But today, this video is on DNA. So I do not talk anything about the uracil that present in the RNA instead of thymine. So these nitrogenous or nitrogen containing bases are associates with the sugars 2'-deoxyribose sugar. Why I called it deoxyribose sugar? We will discuss on that. So the 2'-deoxyribose sugar and the nitrogen base together forms nucleoside. Let's talk about the sugars that present in DNA. The sugar that present in DNA is called 2'-deoxyribose sugar. Why use D? D means deletion. So there is something missing. Notice that in this picture there is something missing. So why it is called 2' sugar? Because there is no hydroxyl at the 2' position of the molecule. Notice on this picture that I highlighted the hydrogen with yellow color in the 2' position holding by the molecule. There is an oxygen atom missing. So this is why it is called deoxyribose sugar. We see that in this picture we numbering the atoms in the prime numbers. Why we use the prime numbers? This because to designate the numbers of the molecules with prime numbers. Why? This is for that reason, to distinguish them from the positions of the nitrogen bases. So there is something to notice, that the 2' position is a oxygen atom is missing. So that is why it is called deoxyribose sugar. And we use the prime numbers to distinguish from them to the bases, nitrogen containing bases also. Yes. So in the second picture we see that the chemical structure of the nitrogenous bases that present in DNA. We can see that there is two bases. They are purines. One is adenine and one is guanine. So we can see that in adenine there is a ring structure. We numbered them 1, 2, 3, 4, 5, 6, 7, 8, 9 according to their positions. And we can see that in the 6th position there is a amino group present. So this is why it is called, I talk about the UPAC name, IUPAC name, UPAC name. So 6 amino purines, the UPAC name of adenine. In guanine there is oxygen atom present in the 6th position, and in the 2 position there is a amino group present. So it is called 2-amino-6-oxypurine, because in 6th position notice that there is a oxygen, and the 2 position there is a amino group present. So this is the UPAC name of the purines that present in the DNA. So let's talk about the pyrimidines. We see the detailed structure. In the next diagram we see that there is two pyrimidines. One is cytosine and the other is thymine. In cytosine, in the 2 position there is a oxygen atom and the fourth position held by a amino group. So this cytosine and its UPAC name is 2-oxy because there is a oxygen atom in the 2 position and 4 position has a amino group. So 2-oxy 4-amino pyrimidine. We numbered those molecules 1, 2, 3, 4, 5, 6. So it is a ring structure. In case of thymine, there is 2 oxygen atom held in the 2 position. There is oxygen, and the 4 position there is oxygen also. But in the 5 position there is a methyl group present. So its UPAC name is 2,4-dioxy because 2 oxygen is present in the 2 and the 4 position. 5-methyl pyrimidine. So this is the nitrogen containing or nitrogenous bases that present in the DNA. So after that I will discuss where DNA exists, where DNA present. So there is no organism without DNA. But in case of some virus, there is presence of DNA or RNA, but not both. Both the DNA and RNA cannot be present in a virus. So we see that there is something. So in the virus, they lack DNA or RNA both. So where present DNA? In the nuclei of eukaryotes like plant and animal cells, DNA is present in the chromosome. DNA is present in the chromosome in association with some other protein molecules. So in prokaryotes, both the plant, animals and other organisms, the DNA is present in chromosomes. With that, in association with other molecules, protein molecules and other molecules. But in case of prokaryotes like mycoplasma, bacteria, in other prokaryotes, there is no such associator is present. So they are not associated with other molecules that we found in the eukaryotes. In the eukaryotes, there is some other associators that present in the earlier we talked about the nucleotides. What is nucleotide? When a phosphate group or phosphate ion joins with the nucleoside, then it is called nucleotide. And the nucleotide is also attached with a nucleoside. Nitrogenous base and the sugar that present in DNA are collectively called nucleoside. Nitrogenous bases are 4 types. We discuss on that. Adenine, guanine, cytosine and thymine are the base, nitrogenous base on for DNA. So the nitrogenous base and sugar, both of them collectively called nucleoside, and nucleoside when attached with a phosphate ion or phosphate group, it is called nucleotide. So in this picture we can see that the 4 bases are shown, and when adenine, a base, nitrogenous base, attached with the sugar deoxyribose sugar, then it is called deoxyadenosine. I told you that when deoxyadenosine means the nucleoside attached with a phosphate group, then it is called nucleotide. So when it is attached with a phosphate group, then it is called nucleotide. The nucleotides are deoxyadenylate. This is the nucleotide for adenine. Followed by the guanine. When guanine attached with the ribose sugar, deoxyribose sugar, then it is called deoxyguanosine. It is the nucleoside for guanine. And when the nucleoside deoxyguanosine attached with a phosphate ion, then it is called deoxyguanylate. Cytosine, when attached with the sugar, deoxyribose sugar, then it is called deoxycytidine. And followed by, it is also attached with a phosphate ion, then it is called the deoxycytidylate. It is a nucleotide. And the last, thymine. Thymine, when attached with a deoxyribose sugar, then it is called the deoxythymidine. And the nucleoside, when attached with a phosphate group, then it is called deoxythymidylate. So another names also another names of those molecules is also present. We can say that the adenosine, when adenosine attached with a ribose sugar, then it is formed deoxyadenosine. But when it is attached with a phosphate ion, then it is also called deoxyadenylic acid. There is I miss those names in the picture, but you can note that. So when deoxyribose sugar and the deoxyribose plus base, that means the nucleoside attached with a phosphate group, then it is called nucleotide. The nucleotide for adenine is deoxyadenylate. In this picture you can show that in the first row. So it is also called deoxyadenylic acid. So there is some names. Shortly it is called DAMP. So in case of guanine, it is called deoxyguanylic acid. In case of cytosine, it is called deoxycytidyllic acid. In case of thymine, it is called deoxythymidyllic acid. So it will be present on the description area. You can go and see those in the description area. And so another diagram. In this diagram, it is clear that the concept of nucleoside and nucleotide. In the yellow ring structure, it is called a nitrogenous base attached with a pentose sugar. Those attachment, when happened, it is called nucleoside. Followed by the nucleoside, the nitrogenous base plus pentose, and the red color circled diagram, it is a phosphate group. When attached with the nucleoside, then it is called nucleotide. So it is clear that nitrogenous base, the four nitrogenous base, that is adenine, guanine, cytosine, thymine, when present, and they will attached with a sugar. For DNA, it is deoxyribose sugar. In case of RNA, it is the ribose sugar. Is for the both cases it is happened. In case of DNA, it is called deoxyribose sugar. It is called the nucleoside. For RNA also it is called nucleoside. And when it is attached with a phosphate group, we can see that in the red color, it is called nucleotide. So friends, that's it for today. I will make different videos regarding DNA and its structure. So stay tuned and keep supporting my channel and subscribe to my channel, and hope you like that. So thank you for watching.